Hey everyone, this is module number 2, module 8, video number 2. So I'm going to post one more, video number 3, but this is what we're going to cover in this video. So last time we talked about mechanisms of mutations and the classification of mutations, and I went through spontaneous mutations, how they occur. So three main categories: chemical changes, natural radiation, errors in DNA replication. Today we're going to start with this: induced mutations, induced by mutagens. And then I want to go over different types of chromosome rearrangements. So these are large-scale mutations, just define what they are and how they come about. So let's take a look at those. First of all, induced mutations. So these are mutations that occur at a higher rate than the rate of spontaneous mutation. And that's because they're occurring in response to exposure to some mutagen. A mutagen, by definition, increases the mutation rate above that of the spontaneous rate. So mutagens are of two main types: chemical mutagens and electromagnetic radiation at high energy or short wavelength types of radiation. So let's start by talking about chemical mutagens, and then we'll finish up with radiation as a mutagen. So I'm going to give you 3 examples of chemical mutagens, and all of these are mutagenic because they affect the chemistry of the DNA molecule, specifically the bases. And so the 3 main types are base analogs, and these are bases that resemble very closely the structure of the normal DNA bases, and they can even be incorporated into the DNA molecule. But when they're incorporated, they're going to introduce chemical instability. Also, there are chemicals that cause direct changes to the chemical structure of the DNA bases. Okay. And then finally, there are mutagens that insert themselves between stacked DNA bases. So these are the 3 types. Let's take a look at the first type first: base analogs. Okay, so here's an example of a base analog. It's called 5-bromouracil. 5-bromouracil is an analog for thymine. So if you feed a cell or an organism 5-bromouracil, and it will be incorporated into cells, and cells will use this to make nucleotides containing 5-bromouracil. And then during DNA replication, DNA polymerase will recognize a nucleotide with this base as a T. And DNA polymerase will insert this opposite an A. Okay? It has the similar base pairing to thymine. It will be incorporated opposite A. So it looked like this in a DNA strand where there's a TA base pair. During replications, these strands will separate, and the bottom strand will serve as a template. DNA polymerase will come along, insert a nucleotide containing 5-bromouracil in the position opposite the A. Okay? Now the thing about this base, 5-bromouracil, is that it undergoes tautomer shift, that is, change in the electron circulation, such that it resembles a cytosine. And this tautomer shift, and I talked about this last time, spontaneous mutations can occur due to tautomerization of the natural DNA bases. But 5-bromouracil undergoes a tautomer shift much more at a higher frequency than the normal bases. And so if this is the form that's present during DNA replication, this 5-bromouracil tautomer form will result in the incorporation of a G opposite to the 5-bromouracil. Okay? So what will happen is eventually when this G strand goes through the next round of replication, DNA polymerase will incorporate a C opposite, okay? So starting out with a TA pair, if the A is replicated and 5-bromouracil is incorporated opposite it, and the 5-bromouracil undergoes a tautomer shift, in the next round of replication, DNA polymerase puts a G opposite because the tautomer version of 5-bromouracil resembles C. Then, in the next round of replication, this G-containing strand will direct DNA polymerase to incorporate a C. You've gone from TA to CG. And so base analogs work in that way. They undergo tautomer shifts at high frequency and will direct base substitution during 2 rounds of replication. For base analogs, they always cause a transition mutation, T pyrimidine converted to C, the other pyrimidine. Okay, so that's how base analogs work. There are also mutagens, chemical mutagens that cause chemical changes to bases. So an example of that is the mutagen called hydroxylamine. If you expose DNA to a hydroxylamine, it will hydroxylate the amino group of cytosine. Hydroxylated cytosine produces this N4-hydroxylcytosine. And this rearranges the electron pattern such that hydroxylcytosine forms 2 hydrogen bonds with adenine instead of the 3 that it would normally form with guanine. So here would be hydroxylamine, hydroxylcytosine going through replication, and DNA polymerase would insert an A opposite hydroxylcytosine. And then in the next round of replication, if the A strand is used as template, DNA polymerase will put in a T. So in 2 rounds of replication, you've gone from CG through hydroxylcytosine to TA. And that's how hydroxylating agents work. And that's how other base modifying agents work as well. They cause substitution mutations, which are transition mutations, C to T, pyrimidine to pyrimidine. Okay, the final type of chemical mutagen is a little bit different than the other 2. And this is a molecule, proflavin is one kind, there are others. But basically it's a 3-ring structure and it's a planar structure. So this resembles actually what would be, the situation would be in a DNA molecule where a purine base having a base like adenine or guanine with 2 rings, base pairing to a pyrimidine base having a single ring. And it takes up basically the same space and it has the same planar structure. And this kind of molecule, called an intercalating agent, can insert between the stacked bases of 2 neighboring base pairs in the DNA molecule. And it would look like this. This molecule is not covalently attached. However, it will remain in the strand during replication. And a DNA polymerase encountering a intercalating agent in a single strand when it's being replicated will result in either the insertion of a single base or the deletion of one of the neighboring base pairs. Okay, so intercalating agents are mutagens that don't cause base substitution. They cause insertion or deletion mutations. All right, let's switch and talk about the other main topic for this video, which is chromosomal mutations. Chromosomal mutations are large-scale mutations that cause rearrangements of chromosomes. And there are 4 main types of chromosomal type mutations, and they're illustrated here on this slide. This illustrates a normal chromosome having the sequence of certain chromosome segments along the length of the chromosome. They're just arbitrarily named A, B, C, D, E, centromere, F, G, H. All right, one type of chromosomal mutation is a deletion, in which a segment or segments are deleted from the chromosome. In this case, BC gets deleted, and you end up with A, D, E, F, G, H. Another type is a duplication, in which a segment or segments are present in 2 copies instead of 1. And if you duplicate BC, you would end up with a duplication chromosome, ABC, BC, DE, FGH. BC segments are present twice. Another type of mutation is an inversion, in which a segment of the chromosome is reversed with respect to its normal orientation. So if this is a normal chromosome and we reverse the BC segment, we would come up with an inversion chromosome, ACB, DE, FGH. Finally, one type of chromosome mutation involves swapping of material between non-homologous chromosomes. And so in this case, you would have 2 chromosomes with normal segment order ABCDE, centromere FGH. And here's another non-homologous chromosome, LMNOP, centromere QR. If you swapped the LMNO segment for the ABCD segment, you would get this. 2 chromosomes having segments in which the original segment is now moved to another chromosome, and in its place is a segment of the other chromosome. So that's called a translocation chromosome. Okay, how are chromosomal rearrangements produced? There are 2 main mechanisms, and they're shown here. One is by creating double-stranded DNA breakage, and usually this is the result of exposure to a mutagen. Radiation is a mutagen. The other is what's called, generally speaking, as aberrant crossing over. And I'll illustrate what I mean by that in a second. To illustrate the first, double-stranded DNA breakage, if, let's say, a cell is exposed to high levels of X-rays, it can produce a number of breaks along a chromosome. And if you have double-stranded breakage here and loss of this segment, and then the A and D segments get rejoined, that would produce a deletion chromosome. Excuse me. On the other hand, if you have the same breakpoints, however, this segment is retained and it's rejoined in the opposite orientation, and you would get ACBD instead of ABCD. That would be an inversion. You could have 2 homologous chromosomes undergoing breakage at different points and rejoining, scrambling of the segments to produce 1 deletion chromosome, AB joined to D, and 1 duplication chromosome, ABC joined to CD. So here the C segment is missing. Here the C segment is present twice. This would be a deletion, and this would be a duplication. You could also have chromosome breakage in 2 different non-homologous chromosomes, and then rejoining to not their original partners. And that could produce a chromosome with AB joined to HIJ and EFG joined to CD. So that would be a translocation resulting from chromosome breakage. So chromosome breakage can produce all 4 different types of chromosome mutations. The other mechanism is if there's aberrant crossing over within a chromosome or between different chromosomes. And aberrant crossing over is abnormal or unusual crossing over. Usually crossing over occurs between homologous chromosomes such that they swap equal chromosome segments. However, occasionally what can happen is this. There are repeated DNA sequences in chromosomes that can line up with each other, and when they line up, there can be crossing over between those repeated segments. And if a chromosome does this, it folds on itself, and these 2 chromosome duplicated regions, repetitive elements shown as the green arrows, align with each other, and crossing over occurs. If you follow this, it will produce A, D. That would be a deletion chromosome. It's missing the CB segment. C and B would be present in this circular molecule, which would be lost in a eukaryotic cell. Or you could have repetitive elements that are in opposite orientation that align with each other. If that happens, you would produce a chromosome now that has ACBD instead of ABCD. That would produce an inversion. You can have two homologous chromosomes that line up out of register with their repetitive elements. Maybe usually A would line with A, B with B, C with C, D with D. But if they align through their repetitive elements here out of register and crossing over occurs, A would be linked to D, a deletion chromosome, and ABC would link to BCD, a duplication for the BC region. Finally, if two non-homologous chromosomes have crossing over between a repetitive element that they both contain, this can produce translocations, AB linked to HIJ, and EFG linked to CD. So these two mechanisms, chromosome breakage and aberrant crossing over between chromosomes, are the two main mechanisms that produce these chromosome mutations. Now, chromosome rearrangements are usually, or can be, large chromosome rearrangements. And these can be detected under the microscope by looking at chromosomes from mitotic cells. In metaphase of mitosis, the chromosomes are compacted to their highest degree. And there are techniques for detecting chromosomes that have undergone rearrangements. So if you look at the karyotype of such a cell, you can do a technique called fluorescent in situ hybridization, or FISH, using probes that are specific for different chromosomes. And then if you shine a laser beam, they will fluoresce at different colors. Okay? So if there's a probe that fluoresces green, that's typically going to detect one chromosome, and a different probe that's fluoresces red, and that's targeted to another chromosome, and you stain a chromosome spread from metaphase of mitosis with these two probes. The others chromosomes are staining with a DNA stain. You will see one chromosome staining green and another one staining red when you shine a laser beam on them. However, you also would notice that this chromosome is partly red and partly green. And this other chromosome is also partly green and partly red. It looks like they've swapped segments of both chromosomes. A small segment of the red chromosome has been swapped with a somewhat larger segment of the green chromosome. So this would be a translocation that you could detect by this FISH technique. All right, I want to go through some examples of chromosome aberrations that can be seen in the main problems that can result. So for chromosome deletions, the chromosome can be either internal to the chromosome, and that's called an intercalary deletion, or it can be from the very end of the chromosome. That's called a terminal deletion. An example of a terminal deletion is the condition in humans called Cri-du-chat syndrome. Cri-du-chat means cry of the cat. And this syndrome produces a number of abnormalities. First of all, just to show the karyotype here, this syndrome is the result of being heterozygous for a terminal deletion of the tip of the short arm of the 5th chromosome. This is an individual Cri-du-chat syndrome. Cri-du-chat syndrome is a dominant condition because it shows up in individuals that are heterozygous for the deletion. That means the deletion chromosome is dominant to the non-deletion. It occurs spontaneously and rarely in 1 in 25 to 50,000 live births. There are a number of abnormalities, including the gastrointestinal system, cardiac malformation, and usually occurs with intellectual disability. The syndrome is named for the malformation of the larynx, which in newborn babies with the syndrome produces a very high-pitched cry, sounding like the high-pitched meow of a cat. So that's Cre-du-Chat syndrome. There are other deletion syndromes, but I will not discuss them this time. This just illustrates the deletion that's present. So it's a small portion of the end of the small arm of chromosome 5. Why is there a phenotype for deletion that's heterozygous? So the situation for deletions is if usually being homozygous is lethal or extremely harmful, and more so for deletions that are longer in length. Deletion heterozygotes can have a mutant phenotype, as we saw for Cre-du-Chat syndrome. And the reason for that is the deletion may expose genes that are haploinsufficient, meaning having only one copy will result in a mutant phenotype. Or the deletion may expose recessive alleles, mutant alleles on the non-deleted chromosome is another possibility as well. For Cre-du-Chat syndrome, the result is due to this, exposing genes that are haploinsufficient. All right, let's talk about duplications, and then one example of that is a duplication of the X chromosome segment in Drosophila, and this results in a particular phenotype called the bar-I phenotype. For this chromosomal rearrangement, it results from an unequal crossing over, so aberrant crossing over between a repetitive element such that a wild-type chromosome having one copy of this 16A region ends up producing a chromosome having two copies. And that produces a phenotype called bar-I, an elongation of the oval shape to the normal wild-type eye. If you have an individual that's homozygous for this two-copy version and an unequal crossing over between that, you can produce a chromosome that has three copies. And an individual having three copies of this chromosome produces an even more extreme version, the double bar-I phenotype. So even more prolonged and narrower. Okay? So the phenotype is a result of the number of copies of this segment of the genome. For duplication or even triplication chromosomes that we saw for bar, the phenotype is a result of change in dose of the number of genes in the duplicated region. And so dosage is important. That's true for deletion chromosomes and also for duplications. How about chromosome inversions? So there are two main types. If an inversion involves the centromere, that's called a pericentric inversion. So this one on the left would be including the centromere in the inversion segment. Or you can have an inversion chromosome that does not involve the centromere. That's called a paracentric inversion. Inversion chromosomes don't change the dose of genes on the chromosome. They just change the order. However, the inversion could produce a phenotype if the breakpoint of the inversion disrupts a normal gene. In this case, if you have a gene and the breakpoint falls either in the gene or between the regulatory region of the gene and the coding region, then that would disrupt this gene, either causing a null mutation or disrupting the normal regulation. So typically, in that case, a phenotype would result. So for inversions, mostly they do not result in an abnormal phenotype, but they could if the inversion breakpoint disrupts a gene, or if it puts the gene in a new regulatory environment. For translocations, there are what are called reciprocal translocations. So these are swapping of material between two non-homologous chromosomes. for a reciprocal translocation, there is no loss of genetic material. And similar to inversions, there would typically not be a mutant phenotype. But, like for inversions, if the breakpoint involved in swapping between these two chromosomes is within a gene or near a gene, this may result in a mutant phenotype. So this is showing the situation for an individual that is homozygous for a reciprocal translocation in which both chromosomes have translocated segments, and so there's no change in dose for any of the genes, and normally this would not have much of a phenotype. Now there is one specific type of translocation that's not exactly reciprocal translocation, and this is called a Robertsonian translocation. And this arises from translocation between two acrocentric chromosomes. Acrocentrics have their centromeres near the end, and if you have a translocation of the long arm of one acrocentric onto the short arm of another acrocentric, and then the other chromosome would have just these two small ends, oftentimes this chromosome can be lost because it has very little or no coding region, no genes. And this chromosome would be retained. So this would be translocation between two acrocentric chromosomes producing one large metacentric chromosome. So this happens some cases, and it is important in evolution. And one example of that is if you look at the difference in chromosomes between the chimpanzees and humans, the main difference is this. Chimpanzees have 24 pairs of chromosomes, while humans have 23 pairs, one less pair. And if you look at specifically what happened, we find that there was a Robertsonian translocation between two acrocentric chromosomes in chimps that formed one large metacentric chromosome, human chromosome 2, in humans. And this accounts for why chimps have two more chromosomes. They would have two of these. Humans have, those two chromosomes are combined. This is known by comparing the sequence of these two chromosomes in chimps to that of the human chromosome. All right, so I'm going to end this video here, and there is one last video to describe mutations that cause changes in chromosome number as well. See ya.